lecture, we're going to get into, beginning the cell membrane. As you've learned briefly, the cell membrane controls what enters and leaves every cell. Every type of cell has a cell membrane. A prokaryotic cell has a membrane. A eukaryotic cell has a membrane. And within the eukaryotic cells, we have plants and animals. Both of them have cell membranes because the cell membrane is responsible for deciding what can enter and leave the cell and really regulating that. And in this lecture, we talk strictly about the structure because understanding the structure of the cell membrane is vital to understanding how it actually functions. So we separate the two and we really break it down, and we're going to talk about it. So the cell membrane is composed of these tiny molecules, well, they're actually quite large, called phospholipids. And if we break this down, phospho, okay, so it's a phosphate group, and lipids, fat. So this is kind of like our cartoon version of a phospholipid. It's easier to draw because drawing this would be quite difficult. So we have this phosphate head here. This is a phosphate group up here, and I'm going to break this down a little bit more. Down here are the fatty acid tails. So phosphate head, fatty acid tails. If we take a look over here, we know that fatty acids from our study in lipids, or lipids, are made up of all carbons and hydrogens, which is why they're hydrophobic. They're nonpolar. Carbons and hydrogens share those electrons equally, which makes them hydrophobic. And it's really important for us to note that the phosphate head is hydrophilic, and I'm going to get into this a little bit more in just a minute. But we take a look here and we see that the structure is bent, and that means it is a saturated fatty acid, which is important. This bend here happens from a carbon to carbon double bond. So that's really important for us to note. Okay. So the phosphate head is what we call hydrophilic. We've talked about hydrophilic before. That means it is polar and water loving, okay? The fatty acid tails are hydrophobic because they are nonpolar, okay? And so hot water fearing, and that actually is really vital to the whole structure. Because of this, what happens is that the phospholipid, our cell membrane, is arranged as what we call a bilayer, two layers, right? So we have this phosphate head up here attracted to water. This is repelled by water. Because of that, if you were to take phospholipids and throw a whole bunch of them into a beaker of water and shake it up, they would arrange this layer. Hold on. They would arrange this layer. So out here is water, water. So you take a look that the phosphates will always arrange themselves to water because they love the water, okay? And our hydrophilic tails are hiding out in here because it protects them from the water. So that's really important for us to remember. That's why the membrane is arranged the way that it is. Everything with a membrane is arranged this way. There's always water outside of a cell. There's always water inside of a cell. So in order to protect the fatty acids from the water, if we think about it that way, that's why it is this way. Okay, so I wanted to break the parts of the phospholipid down even more because it's really important for us to understand what we learned in biochemistry is extremely important. We take a look at this phosphate um, surrounded by oxygens. We know that oxygen is extremely electronegative, and you could see here that it's got these slight charges. So this is the phosphate group that's on the phosphate head, okay? So that shows us, we see this negative charge, we automatically think, oh, that's polar. So if we take a look at what these fatty acids, ignore this for a minute, what this fatty acid looks like, because this is a triglyceride, I just wanted to see the fatty acids. Carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, carbon, hydrogen. These are really hydrophobic. So when we have these fatty acid tails, they're always going to want to be away from water. All right. So we take a look here. So the picture I was just showing you, this is a more accurate model. We have the phosphate heads here, down here, facing the water, the polar hydrophilic heads, and our non-polar hydrophobic tails. It's really important. I really think it's important that you just take a minute and kind of draw this out in your notes. Maybe not this one, maybe this one down here. So we have an understanding of how this is arranged and why it is arranged that way, right? So it serves as our cellular barrier or our border. And what you're about to learn is that it determines, I'm just gonna bring this all up, what goes in and out of cells, right? So it's important to note that polar molecules are not going to be able to go through this way. Sugar is polar, water is polar, salt, highly polar, right? It's going to go try to go through and get repelled right back out. Lipids are going to be able to flow through pretty easily, okay, because they are um, hydro hydrophobic. They are non-polar, okay? So the cell membrane separates the living cell from the aqueous environment. Aqueous meaning liquid environment. It's a really thin barrier. It's about 8 nanometers thick. So 8 millimeters is really small. Now imagine a fraction of that being nanometers, right? We sometimes have a hard time contemplating how big that actually is. It's really tiny. It also controls traffic in and out of the cell. And in the next lecture, we're really going to explore how that happens. So it allows some substances to cross more easily than others. So the hydrophobic nonpolar will get through, especially the very small, and the hydrophilic polar will not be able to get through. Remember, this acts as like a repulsion of those polar molecules. So membrane becomes semi-permeable via protein channels. So we have these protein channels embedded into the cell membrane. These specific channels allow specific material across the cell membrane. So we're looking inside of the cell here, again, with water outside. So we have these proteins, and we're going to really get into this in just a minute, and they're embedded into this membrane. And you should be thinking to yourself, how are they able to be embedded if this is a nonpolar region? That's a great question. We'll talk about it. So sugar has its own protein channel. Amino acids have their own protein channel. Amino acids are big too, okay? Sugar is a somewhat big molecule. Water has very small, what we call aquaporons, and we do talk about that allow it to go through. Salt needs to have its own protein channel, and so does ammonia. So we're looking at some things that can get through the membrane because they have these channels, these little tunnels that they can go through that kind of serve as a protection. So, like I was saying, it's more than lipids. Transmembrane proteins, right? Trans across the membrane, are embedded in the phospholipid bilayer. So we're taking a look at this bilayer membrane, right? It creates these semi-permeable channels. The word semi-permeable is something that we use a lot here. Permeable means able to get through, and this is actually spelled wrong, so I apologize for that. It means they're able to get through. Semi means only certain things, right? So if we take a look, proteins aren't on the top, right? But they are embedded within, and we're about to talk about. Okay, they're in the lipid bilayer. There's a lot of spelling errors in this, and I apologize for that. Okay. So why are the proteins a perfect molecule to build structures in the cell membrane? Think about protein structure. We know that proteins are composed of many, 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 many amino acids, right? A chain of amino acids. And then those amino acids fold up on each other. I'm recording.
Oh, I'm sorry.
Fold up on each other, and they create alpha helix and beta sheets. And then the tertiary structure between the interactions of the R groups. Sorry about the interruption. It happens all the time. And we have these really cool three-dimensional shapes, and you're probably like, oh god, I've heard enough about proteins, but we need to talk about them a little bit more because their structure is important. So we have these different classes of amino acids, and what do all these amino acids have in common, right? What are these have in common? And you guys should be thinking to yourself, oh, we see lots of carbons and hydrogens, okay? These are our nonpolar and hydrophobic amino acids, right? And this is something that we've talked about a lot. The carbon and hydrogens, they share their electrons equally. When you see a lot of carbon and hydrogens, then you know that we're dealing with some nonpolar molecules. But then we take a look at these amino acids, and we have a lot of oxygens. Oxygens are electronegative. We have NH3 plus, we have NH2 plus. We see a lot of charges in here, and we can probably guess what, guys? These guys are polar and hydrophilic amino acids. So all of these amino acids have different charges, and when they are strung together, they can kind of anchor into the cell membrane. So within the membrane, our nonpolar amino acids are going to want to be hanging out in here, and they arrange themselves in that way just because of the structure of the protein. They're hydrophobic, and it really anchors the protein into the membrane. So they have these hydrophobic interactions occurring that kind of keep them together, right? On the outside surfaces of the membrane, because we know that there's water here and we know that there's water here, our polar amino acids are going to arrange themselves. They're hydrophilic, and they're going to extend into the extracellular fluid into the cytosol. So it's really this protein structure. The protein structure and function is a common theme in molecular biology because these guys are so important for so many reasons, and this is just one of them. And because of the way they arrange themselves, it allows for certain molecules that wouldn't normally be able to get through the membrane to get through the membrane. And these are all molecules that we really, really need in our body, right? Okay. So we have an example here. This is an aquaporin. This is a water channel in bacteria, and it shows us the structure of it. It's got some beta-pleated sheets, and we can see that it's arranged in a way that keeps it embedded in the membrane, right? So H2O is out here, H2O is in here. Our polar arrangements are going to be out here. So then we're taking a look at a proton pump. Okay, this is something we talk a lot about in biology. Protons are just hydrogen ions with no electrons. It's a proton, just a proton. And this isn't a photosynthetic bacteria, this particular one. But you're taking a look here. We have our hydrophobic part of the protein, and we have our hydrophilic part of the protein, and our hydrogen ion can go right through. Okay, and that's a really important concept in bio that we end up talking about later on. So they function through conformational shape. Okay, the protein will change shape as these molecules are going through. So there's many functions of membrane proteins. So we're looking at the outside and the inside, and this is the plasma membrane, okay? And we're looking at different types of proteins, and it's important for us to know the different types of proteins that are in the membrane. You have a transporter. I've been talking a lot about those. They act as a channel. You have enzymes that are embedded in the membrane, and we know that enzymes are those things that are speeding up reactions. So you have a substrate come fit, and then the reaction occur, and the product is off to go do its own thing. And so this membrane protein, this transmembrane protein, is able to facilitate certain reactions that are extremely important, especially when we get talking about cell signaling. A cell surface receptor is something that will I'm going ahead of myself. Sorry. Will have something come bind. It will change the shape of the protein, and then a reaction will occur inside of the cell. Again, important for cell signaling. A cell identity marker. This is a protein with a carbohydrate attached to it. It acts as an ID tag. We need our inside of our bodies and all living things. All cells need an ID tag, so they know how to interact with each other. This is an antigen, and I'll be talking about this in just a minute for blood types and everything. I'll just cover it briefly. So we have these two proteins on two different cells. So we're looking at two different cells, and they adhere to each other. Cells being able to adhere to each other is extremely important. And then we have proteins that are just there for the attachment, and that says to the cytoskeleton. And the cytoskeleton is protein fibers that give the cell its shape. Okay. So the proteins are going to determine the membrane's specific functions. The cell membrane and organelle membranes each have their own collection of proteins. So if you remember when we were looking at parts of the cell, and we'll look at them a little bit closer, there are membrane-bound organelles. All of these membranes perform the same way. So the classes of membrane proteins, we have peripheral proteins, and these are loosely bound to the surface, peripheral outside. So examples are cell surface ID markers, antigens. And then we have integral proteins, which penetrate the lipid barrier, and usually they're across the whole membrane. And those are our transmembrane proteins. And examples of that are transport proteins, like the channels and the pumps. So I do want to, okay, so this is an antigen, this is an ID tag, and we're going to talk about that. And then here's our transmembrane proteins. Okay. All right, so in 1972, S.J. Singer and G. Nicholson proposed that membrane proteins are inserted into the phospholipid bilayer. They said it's got to be more than just lipids, right? So they named it the fluid mosaic model. So we're looking at the phospholipid bilayer and our proteins that are integrated along the membrane, and they said it's like boats floating on a lake, right? So then we come up with fluid mosaic model, and we're taking a look here. The membrane is a collage of proteins and other molecules embedded into the fluid matrix of the lipid bilayer. So here is a whole picture of everything going on, and I haven't really talked about what these are. So I'm going to take a moment and talk about this is cholesterol. Cholesterol, we know, is a lipid. It's a steroid, actually, and it is responsible for giving flexibility to the membrane. That's really important for us to understand. So cholesterol is important. It's needed to be embedded into the membrane to allow for flexibility and fluidity. Without the cholesterol, we'd have a lot of problems. So not all cholesterol is bad. I think most of the time we get a bad rap. Cholesterol gets a bad rap, but it's got some really important functions inside of a cell, especially the cell membrane. So we're taking a look at transmembrane proteins. These are filaments of the cytoskeleton. They're like little proteins, right? And we're taking a look, we have a glycolipid here. So this is our ID tags, our glycolipid, also known as our antigens. So just to make note that carbohydrates are polar and hydrophilic, so they're always going to be attached to the outside or the inside of the molecule. And again, cholesterol is embedded in the middle. It's a fat, okay? So the fluid mosaic model. Oh, there we go. We're going to go ahead and label everything else here. Sorry. So there's our cholesterol. Sorry. Take a look at this. I know it may seem daunting, but it probably is important for your notes to take a sketch of everything that's going on here. All the components. We have glycoproteins, we have glycolipids, we have cholesterol, we have phospholipids, we have the transmembrane proteins. Okay, so maybe take a moment and kind of sketch this out the best that you can. I'm not saying you have to be a terrific drawer, but it is something that you need to be able to understand. And sometimes drawing is I say often, in biology, drawing these concepts is the best way for you to learn them. Okay, so I'm going to just talk for a minute about the membrane carbohydrates. They play a key role in cell-to-cell recognition, and that's the ability of a cell to distinguish one cell from another, right? And we call those antigens. It's important in organ and tissue development, and it's the basis for rejection of foreign cells by the immune system. So everybody has a blood type, right? Your blood type depends on what type of antigens you have on the surface of your red blood cells. If you're type A, you have A antigens. If you're type B, you have B antigens. AB, you have both of them. And type O, there are no antigens. But again, these are really responsible in helping our immune system determine what cells are ours and what cells are not. So a lot of times in organ transplantation or even in blood transfusions, we have these rejections because they're not recognizing the antigens. It's really important. Okay. I thought that was cute, so let's leave that in there for you guys. Any questions you have, let me know.